Thank you everyone for taking the time to join this webinar. Today, I'm going to talk about multiple antibody discovery strategies for CAR T therapy development. We will start with a general introduction of a current CAR T therapy R&D landscape, followed by a detailed discussion on the key milestones for CAR lead discovery. For the last part, in order to help everyone better understand the process in antibody lead discovery, we will share a comprehensive case study of single-domain lead generation for CAR T application. Comparing to antibody drug, CAR T therapy comes with a much shorter history. However, it evolves rapidly since its birth. And there have been 4 generations of the structure design within a couple of years. Nowadays, the R&D trending of CAR T therapy lies in 3 aspects: efficacies, diversity, and compatibilities. For starter, efficacy has been the first priority for the last decade. Multiple activating domains are engineered to be incorporated into the intracellular region to send a positive regulatory signal to extend the lifespan of a T cell, to induce the T cell activation, or to induce the cytokine release. In the meantime, safety is another major aspect within the CAR T design. Different strategies has been employed to lower the risk of a cytokine release syndrome. In terms of diversity, after the first wave of pipelines that focused on hematological tumors targeting CD19, BCMA, people start to shift their attention to solid tumors targets such as GPC3, HER2, and mucin-1. And the different designs are also introduced to help the T cell to better infiltrate the tumor microenvironment. For the compatibility, the major pain point of a current CAR T product is that it has to be generated on a patient-by-patient basis, which leads to extended timeline of a drug manufacture and extremely high costs. Off-the-shelf universal CAR T is the solution that aims to improve the flexibility and the cost saving for future CAR T therapy. Currently, there are 3 products officially launched, 2 from Kite and 1 from Novartis, followed by more than 600 ongoing CAR T R&D pipelines all over the world. In addition, over 40% of them are targeting CD19. How do we make ourselves outstanding among such a competitive and crowded field? To put it in a simple word, 3 major aspects can be considered. First is new target, such as those on solid tumors. The new modality, including both single-chain FV, a single domain, or bispecifics, and a new lead discovery strategy to expedite the R&D process. Here, I'm showing a general workflow of CAR T lead discovery. During the target identification stage, not only the target itself, but also a panel of key parameters needs to be decided, such as desired affinity, specificity, cross-species reactivity, etc. These will play a crucial role in determining the detailed strategy, screening criteria, and the validation matrix for the subsequent lead generation. After obtaining desirable lead candidates, either in the format of a single-chain FA or single domain, the CAR is designed and constructed, followed by various packaging for CAR gene delivery. The CAR T cell will be validated by in vitro cell-based assay and the in vivo animal model before the R&D enabling work and the clinical trial. Next, let's move on to the next section, the key milestones and the typical strategies for CAR lead discovery. Currently, CAR antibody generation practically utilize the same platforms as therapeutic antibody. At GenScript ProBio, 4 different platforms are commonly used: hybridoma, single B-cell screening, single domain naive phage library, and immunized phage library. Although sharing the same technology, the detailed discovery strategy is still different for CAR T applications. For therapeutic antibody, function and efficacy is a crucial factor in the decision-making process, and people tend to position the functional validation at earlier stage of a candidate screening. Meanwhile, the overall scale of a screening is also relatively larger to increase the chance of success. However, for CAR antibody, its function is achieved by T cells, not the antibody itself. As a result, people usually don't carry out much functional screening until lead validation stage. On the other hand, affinity and specificity assessment is moved up in the chain of a process. Moreover, the overall scale of a screening is much less than drug discovery. To break it down, the first key milestone is lead sequence generation. In order to better define the specific strategy of a lead discovery, we usually start with the following 3 questions. First, which modality should I choose? Single-chain FV can be easily converted from IgG. This feature makes it more popular in the field, since there are great flexibilities in terms of platforms and the strategy for IgG lead generation. However, this conversion usually comes with affinity loss, sometimes by the range of 10 to 100-fold. Alternatively, single domain is a naturally generated antibody with retained affinity. In addition, the size of the single domain antibody gives its unique advantage for certain inaccessible epitope. The second decision is the origin of lead. Typically, antibody from immunized animals comes with better affinity, but also the risk of immune tolerance and takes much longer time for the whole process. Alternatively, lead generation through naive or synthetic library showed absolute advantage of turnaround time, but at the cost of a potential low affinity. This brings the third question. Is affinity always the higher, the better? Since the major function of antibody in CAR T is target engager, desirable affinity and specificity is a must-have. However, considering the potential adverse effects of activating T cells, extremely high affinity might not suitable in every circumstances. Actually, in some of our case studies, antibody with the affinity of a nanomolar range also showed pretty decent functional efficacy. Here, I listed all 5 different strategies for CAR lead generation for the side-by-side comparison, including timeline, costs, pros, and cons. For the single-chain FV format, hybridoma is the most cost-efficient way, but takes longer, while naive library is the most expedited approach. Single B-cell based screening is the most expensive one, but comes with a better diversity. For the single domain format, immunized library is more suitable for target with high affinity requirements. Here, I'm showing the general workflow of a single chain FV lead generation with 3 different approaches. Single B cell based technology isolates B cells from immunized animals and identify top leads through cell level screening, sequencing, and affinity validation. Alternatively, hybridoma strategy takes the splenocytes from immunized animals to generate hybridoma cell line to facilitate the screening, subcloning, validation, and the sequencing. Lastly, human naive library takes a total different ways by phage display panning, screening, and sequencing, plus affinity validation. In terms of single domain lead generation, there are also 2 different approaches by similar phage display platforms. One is to utilize the PBMC from immunized llama or alpaca, while the other takes the unimmunized animals as the source of antibodies. Once the library is constructed, the rest of the panning, screening, sequencing, and the validation process are essentially the same. Once the lead candidates are identified, the next step is sequence optimizations. Typically, the following 4 parts of work will be considered for therapeutic antibodies. However, it is not necessarily the same for CAR lead antibody. Humanization usually is a must-have since it can greatly reduce the overall immunogenicity with relatively low costs and risk. For affinity maturation, it depends on the actual affinity and the target requirements. As I mentioned previously, we have observed that nanomolar range of KD is acceptable, but it may need picomolar range for certain targets. As for immunogenicity assessment, it is generally not considered worth doing at the discovery stage. CAR-T injection is much less frequent than antibody drugs, and it is commonly evaluated in vivo instead of in vitro at a later stage. For the developability assessment, it is no longer needed since it is in vivo expressed and the people don't have to worry about its stabilities, purities, and all sorts of biophysical and biochemical features. For humanization, the most popular strategy is still CDR grafting plus back mutation for both single chain FV and a single domain antibody. Typically, people will start with selecting a human IgG acceptor by ranking of homology. Then CDR grafting will be conducted to replace the majority of a non-human backbone in the IgG sequence. The replacement of a framework usually leads to decreasing affinity due to the change of a ribose region structure. As a result, homology modeling will be applied to help design back mutations within framework region. Different back mutation combination will generate a panel of candidates humanized sequence. And affinity ranking tools such as SPR, Octet are commonly used to assess the effects of a back mutation and screen for best candidates with minimal affinity loss. If the lead antibody is not ideal in terms of affinity, in vitro antibody maturation by random mutation within CDR region is a common strategy to improve the affinity. Considering the number of possible combinations of a random mutation within all 60 residues within CDR, we recommended a strategy called two-step precision mutation library. So basically, a single saturation mutation library for each CDR residue is first constructed and screened by antigen binding affinity ranking. Then the beneficial mutation that can improve the off rate identified in the first step will be built into a combinatorial library to screen for the best combination of a mutation, contributing the most to the affinity. Once the lead sequence is finalized, we will move forward to in vitro functional validation. Typically, three kinds of cell-based assays will be considered. The first is cytotoxicity assays, measured by either LDH release from the lysed cell or the luciferase from a reporter cell. The second type is cytokine release assay, and it reflects T cell activation by measurements of cytokine release, such as interferon gamma, TNF, and interleukins. The last kind is T cell proliferation assays by CFSE staining. Here I'm showing a general workflow of CAR-T functional validation using in vitro cell-based system. CAR-T cells are generated by isolating PBMC from human donor and transfection with CAR encoding lentivirus. CAR expression level is confirmed by flow, followed by cell-based assays to validate functional efficacies. Next, I will share some data from a single domain antibody lead discovery internal case study to help everyone better understand the general process of CAR lead generation. We started with single domain immunized library. After several rounds of panning and ELISA fax screening, we obtained more than 100 unique sequences. Then based on the sequence homology and the cluster, we selected a few representative candidates from each cluster for recombinant production and affinity validation. The top 4 clones showed picomolar range of KD from 10 to the minus 10 to 10 to the minus 12. In the meantime, we also conducted panning and screening using the naive library approach. 67 unique sequences were obtained from the naive library, and the affinity of top leads reaches single-digit nanomolar range. After reviewing the affinity and the specificity, we further selected 1 leads from the naive library to conduct the humanization using the strategy described in previous slides. Most of the humanized candidates maintain the original affinity at 10 to the minus 9 range, which is a single-digit nanomole. In addition, we also selected another leads from immunized library to carry out affinity maturation by the approach as previously introduced. The affinity improved 6-fold and reaches 10 to the minus 11, which is almost the upper detection limit of SPR. After sequence optimization, we selected 1 finalized sequence and constructed the CAR T cell using the 2nd generation CAR design. Flag tag and CD3 biomarkers were used to confirm the expression level of CAR domain. Based on the FACS results, the flag and the CD3 double positive cell population is roughly 46% as compared to less than 1% before the transfection. After expression confirmation, both luciferase-based killing assay and cytokine release assay were conducted to evaluate the in vitro efficacy. In the cytotoxicity assay, the CAR T cell showed roughly 10-fold enhanced cell killing compared to non-transfected cells. In the cytokine release assay, we observe a similar trend that the CAR T cells demonstrated 6-fold increase of interferon gamma induction level. Now, for the last part, let's have a quick summary of typical strategy for CAR lead discovery. If high affinity is necessary, immunization-based approach is recommended, either by well-developed platform like phage library or hybridoma, or new technology with shorter timeline like single B-cell screening. Alternatively, naive library or express immunization can be a good solution to expedite the process. If nanomolar range affinity is acceptable. As for modality, both single chain FV and single domain are proven to be viable option with each unique advantage. Now, before I end this session, I would like to give a few words about ourselves. GeneScript ProBio offers a comprehensive platforms of one-stop solution for biologics discovery, including lead generations, lead optimization, in vitro, in vivo assays, antibody characterizations, and druggability evaluation. For lead generations, we are probably the only CDMO in the market that offers 3 different major approach, including hybridomas, phage display, and single B-cell screening. As a professional CDMO, we have more than 10 years of experience in therapeutic antibody discovery. And so far, we have successfully delivered more than 800 discovery projects. In addition, we have signed off 11 therapeutic antibody licensing deal with 6 global biotech and 3 strategic partnership with 3 major biotech companies for GeneScript SMAP body bispecific antibody platform. Our strength lies in the integrated and innovative platform for acceleration of drug discovery. Thank you everyone for your attention. Let's move forward to Q&A session. Before I answer your questions, here is a list of common ones that we collected from previous presentation. The first one is, can customer order this service with already established lead candidate sequence? Yes, definitely. We can take it over from the middle. It's not necessarily all the way from the beginning. The second question is, do we have a recommendation regarding platforms or strategies? As mentioned in the presentation, single domain naive library is probably the one that we're going to recommend. However, it still depends on the particular targets and specific performance requirements from clients, from the pipeline itself. Lots of cases show that budgets and the timeline plays a role in this decision-making process. But in general, we're going to recommend the naive single domain library platforms. The third question is, do we have a QC procedure that ensures the antibody do not misfire and target other cells? Currently, our common strategy to address this issue is that client provide us with a list of a potential off-target antigen or cell lines for evaluation. Then we will do some validation work to see if there is any cross-reactivity against those targets. We do not have any antigen library ready for these off-target screening, but I do believe there are commercially available one on the market. The last sample question is, how many lead candidates is considered as enough for early stage binder discovery? Antibody discovery is essentially a number game. It is always the more, the better. However, considering the cost and the benefits, usually 10 to 20 validated candidates is enough to move forward for functional characterizations. Okay, now let's take your questions.